All right, hello everyone. This is lecture 12, and we're going to cover genome annotations. And we're going to pick up where we left off last lecture with biostrings and move from there. But before we go there, I wanted to talk a little bit about um, functions in R. And um, you guys already know functions. You've already been using them since the first class, basically. But you've been using other people's functions, and you can actually define our own functions. Before we go on to do the technical thing, I'd like you to think about what the purpose or value of a function is. So it's a way to encapsulate code so that it can be executed or run over and over again with different arguments. So imagine we didn't have the function for a median. Then every time we wanted to compute the median, we would have to write down the code to compute the median of a vector of numbers. Instead, we don't need to do that, of course, because the base package of R offers a function called median, and we know that if we pass it as pass a parameter to that function that's a vector of numeric or integers, etc., it will successfully compute the median. In general, what functions do is encapsulate code that make it, in a way, it makes it much more understandable and much shorter. So if every time we wanted to create a scatter plot, we actually had to sit down and write the code to determine the points on the screen, their shape, their color, their size, the axes, the labels, everything else that we know about a scatter plot, it would be a lot of lines of code. And if we wanted to create another function that used slightly different parameters or slightly different data, we would have to copy all of that code again, and that's clearly wasteful. And if we were to do that, our codes would be huge, right? Instead, what we do is we encapsulate that code that does a specific function, like creates a scatter plot or computes the median, in what in computer science they often term a black box. Now, the reason they call it a black box is that we don't really care what happens inside this black box as long as it works. In other words, and we've been doing this all through the course, right? We go and we look on Stack Overflow or in the help manual or anything else online, and we find functions that seem like what we need, and we try to understand the parameters that we need and the that we need to send, right? What data we need to send to that function. And we get an idea from that help manual of what the output of that function should be, either a plot on the screen or a single number that represents the median, etc. So we don't really care how ggplot lays out that plot on the screen, all the calculations that go into it. Likewise, we don't really care how the computer scientist implemented the median function. We have to sort of trust that it works, in other words, right? We assume or we trust it, or that it works, or we try to validate ourselves that it's working. Okay, and that, you imagine if we were designing software for a driverless car, or an airplane, or a financial system, or complicated health information software. If we didn't have functions, it would be almost impossible to debug. The code would be so long and unruly. Functions are a way of pigeonholing our information in nice ways, as you, I think, intuitively know already. They're really the entry point to a field called software engineering. And software engineering is basically the science of designing large information systems. Okay, well, we can define our own functions, as I said, and here's a quick shot at it. Okay, and there's a number of things I should explain in here. My reverse is a variable, just like any other variable we've seen in R, but its class is going to be function. And so my reverse is assigned equal to the result of assigned equal to this function here. Now, this function, it has certain parameters. The first one is called target, and the second one is called upper. And the idea here is that this target's going to be a string, and my reverse is going to reverse that string. If upper is equal to true, then it's also going to convert that string to uppercase. Otherwise, it'll leave it as it is. Okay? All right. So here, upper, I put here equal to false, and this is called the default setting. So if I call my reverse without upper defined, the default value is going to be false. It won't convert the target to uppercase. And you've seen lots of examples of these defaults already. Sometimes you leave out na.remove, and other times you put it in explicitly. And when you've left it out, what's happening is that na.remove is probably equal to false. But it can override that by saying na.remove is equal to true when that's the case. So you've seen lots of examples of these kinds of defaults behavior before, where you don't need to specify all of the arguments to the function. Okay, now the second thing here is these curly braces, okay, that go from the end here to here. And these curly braces in R denote a body of code. So you can think of this as being sort of an atomic unit of code now. Because it's between those curly braces, it'll be sort of executed as a chunk. Think of it as a chunk in R Markdown in a way, right? It's just a group of statements inside, and function calls inside, that are associated now with the function myReverse. Okay, so anything that's between this closing and opening brace belongs to the body of the function myReverse. Okay, so then what does my function myReverse do? It starts off by using the string split function in the tidyverse for my target, and the pattern is equal to an empty string. That's denoted by two quotes with nothing in them. Let me show you what it does. If I come over here, I could say t is assigned equal to ACCGT, arbitrary string, and if I can say TT is equal to string split of t, and my pattern is equal to empty. What I get back now is actually a list and a vector, and inside the first element of the list, that would be, we all know from list now, that's t1 like that, it's double square brackets. You can see, well, I'm sorry, I meant to say this is tt, double square brackets is ACCGT, okay? So, in fact, in here in my code, you can see that target is assigned equal to string split of target and empty pattern, and I just take the first element. Why? Because target in general could be a set of targets, not an individual one. And so it uses a list to give back the results from each element of that list. But here, we only have one. So what I'll do is I'll say TT is set equal to the first element of the list that's returned. And so TT is this broken up like this. And it's just convenient sometimes to work with strings this way. So now if I go back to my my code, my little picture, let's see here. The second thing this function does is reassigns target. So target is set equal to target from the length of target down to 1. And this is what reverses the string, of course. Instead of going from 1 up to the length of target, I go from length of target down to 1. So if I come in here, I can see again, if I take, I can say TT, 3 T's is set equal to TT from the length of TT, which is going to be equal to 5, right, down to 1. And now if I look at triple T, it's reversed the string nicely for me. Okay, so now I want to get it back into a normal string format, so I can use the string concatenate function in the tidyverse. So now I could say TTTT is set equal to strings underscore C triple T collapse is equal to collapse with nothing, and that should give me, yeah, my reverse string as I wanted. Now the last thing here is to check whether upper is equal to true or false. Now by default it's false, but if I had passed true, what it does is target now is set equal to string to upper of target. That does exactly what you think it might, that it should do. And finally, Now you can see here, this is an if statement, so I should explain that too. We've seen a little bit of that now, but not too much. This is a conditional, so this is really, you know, well, that second property of modern computing, which allows us to react to the environment and make decisions. So here we're saying if the variable upper is equal to true, then I convert target to uppercase. And so I reassign target set equal to the upper of target only when upper is equal to true. If upper is not equal to true, it never enters this body of code. So once again, you see the curly braces here being used to denote a body of code that in this case is associated with this if. In particular, this conditional here. If that conditional is true, the code inside of that first body will be executed. If this is not true, if this condition is false, it'll be skipped over. And finally, we use this special function called return, which basically returns the value from the target. So if I go back to my image up here, the return is what forces this black box to spit out the result of the function, in this case target. Okay, so if I go over to my code here, I can see that that's the same function I have on the other page. I can copy and paste this in. And you know, one thing here is I can just say my reverse without any parenthesis, and I'll see now my code from that my reverse is this function with this code in it. So in fact, I can ask the class of my reverse like that, and it says it's a function. And now I can call my function. First, I'll just call it with ACC, this string here, and that's going to match it to this parameter here called target. So when I do that, I get back in lowercase TAGCTAA, the reverse of this guy here. And in fact, if I wanted to convert it to uppercase, I can now explicitly say upper is equal to true, and you can see that it's now in uppercase. If I name my parameters, the order doesn't matter. So here, if I explicitly say that upper is equal to true, then it fills in that this guy is this, and this guy then must be this. So it tries its best to figure out what you mean. If I type in here, I should get the same thing, which I do. And then, well, then one question is, what should happen here? If I copy and paste this in, what's the how is R going to react? And that's pretty hard to predict. I didn't get it right when I thought about it. It's amazing. R always tries its best, and here it didn't actually give an error. It just tried to reverse the word true as EURT, which is pretty strange behavior. Yeah, it's just another example of R trying to make the best out of whatever situation it's given. Of course, you know, we intended that this to be the Boolean, the logical that sets upper, but since we didn't name our parameters and the order is different, well, it had no chance. Okay, so that's one small little function, but we're going to write some more functions as we go along in the course. They're handy, and of course they keep your code succinct, because, okay, to go back then to the very beginning, imagine you don't have this function my reverse, right? And if you want to do this string, okay, so you have to write out the code, all of this code, all of this code has to be, well, without the function, this would have to be copied, you know, here to do that. And if I even change this string a little bit, you know, I would have to then copy all of this code again. And that's not a very smart way to proceed, of course. Okay, so now let's return to our discussion of genome annotations. Okay, we're back. In fact, this is the last lecture in the second part of the course. This is the last lecture on bioinformatics. And after this, we'll be moving into computational biology and looking at some modeling. Okay, so biostrings, yeah, it's a nice package for genetic strings. The main thing is that genetic strings, like genomes, are very large, and they require special functions for them. So, we'd like to be able to compute, for example, the reverse complement of a DNA string. Okay, so here, recall that Biostrings has different types of objects. So one would be the DNA string, and we could define it like this. That's definitely nucleic acids. Is assigned equal to DNA string. Here I've used the equal sign. I don't really actually like using the equal sign. I think I've said this a few times now. It's safer to use the assigned equal to. And again, the problem is that because this means a test for equality, and that's very different than this equal, and sometimes people make mistakes, but it means the same thing. So you can see that R is recognizing this now as something of a DNA string object with 12 letters. I can ask the length of a DNA string, okay, 12. And then, like I said, there's certain functions you'd like to be able to compute on DNA. For example, the reverse Watson-Crick complement. So you can see now that this is TTC for this AAG, right? So it's been reversed and complemented by the Watson-Crick code. And we might believe in translated DNA string. So we would take this codon, this codon, this codon, and this codon, and they code for MIKE respectively. Okay, it's kind of convenient to have around. Okay, so and there's a bunch of other functions on DNA strings, then there's other data structures like amino acids and RNA that seen here. For example, here this is an AA string now, and they have also certain specific functions that make sense for them. The second thing was this BSgenome. We used this package last class a little bit too, I believe, and the nice thing is that they allow sort of a seamless import of full genomes into R. And I think that they derive from the UCSC genome browser. The naming convention is always the same. It starts off with BSgenome, and then the specific organism like S. cerevisiae or Homo sapiens, who did it and what version. So for example, if I want the yeast genome, I can just load this library, and now I'll have this object called S. cerevisiae with several different attributes, including chromosome 1, like that, and I can assign that equal to yeast 1 just for a bit simpler ease of referencing to it, less typing. And I can see that it's quite long. It's 230,000 base pairs long, and it starts with the C and it ends with a, move this over. It ends with a G, and in between there is some 200,000 nucleotides. So, and then there's functions on that Biostrings allows on these objects, like yeast 1, which is a DNA string. For example, I can compute the relative probabilities of each pair of dinucleotides, everything from AA to TT, and you can see that, well, this is 0.1, and some of them are quite small, 0.03, 0.03, and some of them are a bit larger, right? 0.1 I think is the largest. Well, actually this one's even larger, TT. Okay. So that's just convenience, right? That you have these functions that allow you to compute kind of typical things you would on molecular sequence data. Okay, so we have DNA strings, RNA strings, amino acid strings. I'm not going to go through them all here. They all have their functionality. They're all basically different types of biological strings, or B strings. Okay, so B strings is the parent class in some extent of those three specific classes. Again, I can have like an RNA string. I can use the RNA string function on a DNA string, for example, the one that I had before, and now I see that it's translated to RNA. The theta becomes U's, and I could, for example, I could assign an amino acid string like this, and that star means the end, stop codon. And I can test through the class, and I can see that's an amino acid string. Okay. That's pretty straightforward, I think. In fact, the BSgenome series is quite large, not huge. Most of the standard model organisms are there. I think, yeah, this is B. I don't know what Alirata is. That's cow, worm, fly, yeah, fish, I believe, Dan Rio, yeah, E. coli, chicken, so there's a lot of different mouse, rat, all the main, what do you call them, model organisms are there. And for example, if I load the human genome, I can see that I get some information that's coming from the UCSC. It's HG19, we talked about that last class. And then you can see that we have a bunch of chromosomes. I mean, I'm not sure exactly what all these guys are. I mean, this KB things, they're probably fractions of a chromosome that haven't been placed yet, so that data's there. But I can ask, well, I can print out the human chromosome 22, and well, this N, I'm not sure we spoke about this last class or not, but N means that the nucleotide has not been determined. So both telomeres actually of chromosome 22 in human, they haven't really been sequenced where they've been able to determine non-ambiguously what nucleotides they are. It's likely because they're highly repetitive regions, and the sequencers don't do very well in those kinds of cases. Okay. Again, there's these functions like alphabet frequency that I can check the nucleotide usage per chromosome. In this case here, it's gone up to chromosome 22 plus X and Y and the mitochondrial chromosome of human. And then all of these kind of strange chromosomes that are likely incomplete somehow. We can kind of ignore those. And then I compute the frequency of A, C, G, and T in each of these chromosomes. And you could say, for example, compare across chromosomes to see if there's any big differences. Before, you can see already the mitochondrial genome is quite different than the other chromosomes. And well, it's a good exercise for you is to understand the S-apply function. We haven't covered that in class, and it's a bit tricky, but see if you can understand it. Here, you need to, Homo sapiens that object has at least, 22 or 23, 24, plus all of these guys down here, different chromosomes. And so what S-apply does is essentially iterate over all of those different chromosomes and uses this function. It's kind of an internal function to S-apply. And for each such chromosome, it computes the alphabet frequency, and these are just parameters to that alphabet frequency. And so for each seq name, in other words, for each chromosome, it will compute, for example, here, the frequencies in chromosome 1 all the way down to the frequencies on the mitochondrial chromosome, etc. Now, a really important idea in computational biology and data science is pattern matching and alignments. And we're going to come back to this a lot in the second part of the course, in different contexts at least. Next class, we're going to look at a type of pattern matching based on position weight matrices for transcription factor binding sites. But the simplest form of an alignment is something like this match pattern function in biostrings. So basically here, you give it a pattern like this. Okay, that's a nucleotide, I don't know, about 10 nucleotides in a row. And you ask if that pattern can be found on chromosome 2 of the human genome. And what you get back from that is something like this. And it says that, based that here, chromosome 2 is big. I think it's something like 243 million base pairs. And this pattern shows up at 3 million base pairs, 3.09 million, to this number here, which is just a little bit downstream. It's quite short, right, from 68 to 79. and that's 12 bases, and it also shows up a little bit downstream from that at around what is that, 3.9 million, at 4.6 million, at 4.9 million. So it shows up like 89 times in the human genome altogether. So that's kind of nice, because now you've searched over the whole genome for that motif, for that little sequence. If you have, for example, this V-match pattern, it's similar, but I'm sorry. We didn't search over the whole, with match pattern, we didn't search over the whole genome. I misspoke there. We only searched over chromosome 2. Now, if we wanted to search over the whole genome, we could use the V-match pattern. It's just a V. It's the same thing, but now this is a set of DNA sequences, as we discussed on the previous slide. And so now you can see actually that it showed that motif is present 2,300 times across the whole complete genome of human. Okay. Now, there are a lot of different notions of alignment. So here's another slightly more general form. This is an example of a regular expression. And here, my restriction enzyme pattern, PPI1, it needs to have a GAAC at the beginning, and it needs to have a CTC at the end. But in the middle, it allows any nucleotide. That N means any nucleotide. So this is going to search through the genome for all those regions that start with GAAC, and then they don't really care, but as long as the next 5 nucleic acids is anything, and then it has to be followed by a CTC. And so if I ask for this restriction enzyme site in the chromosome 1 of humans within this window of 17,000 to 23,000, I'll see it shows up just once. I think it's just once here at that position, 5,695 to 12, well, whatever that is. I guess it's around 12 base pairs later. Okay. So, oh yeah, right. So here you see it's GAAC, and here it's CTC, and it doesn't really care what they are in there, but you can see that in here it's CCACA. And then there's even more biologically relevant notions of alignment. And those notions of alignment are non-trivial to explain. They're based on models of evolution, of PAM matrices, and nucleic acid mutation models, Jukes, Cantor, etc. And we're going to talk about those really soon in part 3 of the course. But for now, just to give you the intuition, I can ask for a pairwise alignment in biostrings. And this is my one fragment here, okay? And I'm aligning that to this fragment here. And the goal is to find if this string here, the pattern is somehow located in this guy here. But it definitely still wants to match exactly, but there's different things that can occur. You can have mismatches, and you can have these gapping events you see here and here. So this AC matches perfectly with this AC. This ACT matches there, but that T is a problem, and that's why we put a gap beside it. Same thing, this whole region here matches perfectly, and this region matches perfectly, and this one, but I've had to insert these gaps to get everything to line up appropriately. And that's closer to the real notion of alignment that we use in data science. Although there's many things, and I'm not sure we're going to explore them all in this course, that's for sure. For pairwise alignment, the one that most people know is BLAST. It's the industrial strength one. It's supported by the NCBI, and we are going to look a little bit at BLAST downstream. Okay, so that's basically biostrings is a way of representing biological strings, and BSgenomes is one way to bring genomes quickly into R. But even once you have the genomes represented as biostrings in R, there's still the need to annotate those genomes, right? They're not really very useful if it's just a long nucleic acid sequence. really what you want to do is know where the genes are, where the exons are, the introns, the UTRs. You want to know where the binding sites are in promoters, where enhancers are, retrotransposons, all these kinds of objects you would like to know. And you would also like to know things like where there's polymorphism. So, for example, I don't know how many of you have heard of the 1,000 Genome Project, but as you can guess, the 1,000 Genome Project, let me get it up here, was an attempt to sequence 1,000 humans. I don't think these humans were sick in any way. I think actually were several thousand humans in the end, but they come from all over the world. I don't think there's any from Canada actually, but you can see, so that would cover all of our race and ethnicities, I guess, or just race, I'm not sure. But so the idea was to get a kind of a global picture of what human genomes look like. This wasn't a disease study per se, it wasn't a behavioral study. This was just a basic science study of understanding how do human genomes vary, right? And they can vary in different ways. One is a polymorphism, okay, or more formally, a single nucleotide polymorphism, okay? So, as I say here, we call them SNPs, SNPs, or SNPs, single nucleotide polymorphism. So, a SNP here, for example, this might be the kind of the reference human genome. But I might, instead of having a CG dinucleotide here, I might have TA instead, because maybe that controls hair color or eye color, or it controls balding, or it controls nothing, or it could be junk, it could be any different number of things. And generally speaking, these are studies of how our genomes differ. Of course, are very large and very what you call ubiquitous, I guess is the right word. There's thousands of them that are ongoing right now. They're commonplace these days. On just basic biology projects, we could there are many GWAS studies that are associated with disease, that look for associations with disease, and this is a nice paper to explore. Where did it go? There, it's coming. Yeah, this is from just 2019. It's a paper that looks at risk factors of breast cancer, so specific SNPs that put women at higher risk of developing breast cancer over the course of their life. These would be, for example, genes like BRCA1 and 2, which are old markers or well-known markers of breast cancer, and they're looking for additional ones through large genetic, what they're called GWAS studies, genome-wide association studies. Okay. Let's go back here. There. And it could be studies, GWAS studies, that look into human behavior. For example, this was a controversial article that came out maybe a year or two ago, maybe 2019, that looked at genome-wide association studies for SNPs that influenced sexual behavior. And well, as you can imagine, there's it's a sensitive topic, but this is one of the largest studies, and yet still it was heavily criticized for its design. What they found was complicated but quite interesting. So essentially, I think it's safe to say is that they found that there are certain markers, that certain SNPs that do associate with sexual preference, but they don't explain all of the variability. So that's what a lot of geneticists like to talk about, is how much each individual polymorphism explains of the observed variability in the population. And so if you were to put a number on it, you would say that between 5 and 20% of our sexuality is genetically determined. But of course that means that the majority is not genetically, it's not at least obviously how it's not obvious how it is genetically determined. It doesn't mean that there's not other factors there, but it would definitely seem that our sexual behavior is influenced both by our genetics and other factors that might be environmental or social or psychological. So that's been a well scrutinized paper from many angles. But I think those things are quite interesting and potentially very informative. Okay, so where did my window go now? And I can close this. And I should have my, yeah. Okay, sorry about that. Yeah. Okay, so we can bring those. There are tools out there for bringing SNPs into the annotations of these human genomes. There's in this genomes, there's a function called available SNPs. It tells the different studies that are available, and you can load them just like you would a BSgenome into your session, and you can start to ask, and you can map those SNPs or inject them, as they say, onto the human genome, so that you can see that on chromosome 1, for example, that there's 1.89 million, 1.849 million polymorphisms across that whole. That's a very large chromosome, though. Chromosome 22 is the smallest, and there's still some 331,000 mutations. This, I think, was the 1,000 Genomes project that they're coming from. So there's lots of variation in our genomes across the planet. Okay, so yeah, and you can start to dig in as to what those individual polymorphisms are. Are they in genes? Are they in introns? Are they in wastelands that have no coding potential whatsoever? Are they, if they're in an exon, do they change the amino acid? Are they non-synonymous? In other words, are they synonymous, etc.? So, and if it's non-synonymous, is it on the cell surface? Is it, in the protein surface, or is it buried in the protein core? Does it change the chemical property of that amino acid? Like for example, switching an isoleucine for leucine versus changing an isoleucine for proline, two very different events. And will that affect the function of the protein, etc.? There are a lot of annotation packages, and we're not going to cover them in this course. They're very specific to the type of biology that you're doing. If you're into epigenetics, like my wife is, or into genetic studies, this Annotat R is very popular right now. That helps you both, import data, but also visualize it with these kinds of use. Oh yeah, right. So, and then there's RTrack layer. And this, if you've seen the UCSC genome browser, you know that there's different tracks, as we've seen, for different kinds of information. And RTrack layer allows you to add tracks to the UCSC genome browser. You can, in fact, run the UCSC genome browser locally, and you can, inject or project your data onto the genomes. In general, you have all sorts of dependencies between these packages. Like we looked at Biostrings and BSgenome. RTrack layer is the next level where we annotate genomes and make that available. On this side here, it's about RNA sequencing tools, and we might come back to that later. So they're nice packages. It takes a little bit of time to get used to them. But for the purposes of this course, we're not going to get into those details. I want to show an example with S. cerevisiae, or baker's yeast. This classic organism, right? It's like a toy organism in essence. It is a small genome, relatively speaking, just 12.1 million base pairs. It's eukaryotic, it's fungi, of course. It has 16 chromosomes and one mitochondrial genome. It only has about 6,200 genes, so it's much smaller than the human genome. Here's a picture, electron micrograph of a yeast population. So, this allows us to download the genome completely into our R sessions. If you're working in the human or most genomes, then it's a little bit more complicated. You need a pretty good machine to store 3.6 billion base pairs of the human genome and bring that into memory. I should actually say that BioStars biostrings, and this BS genome, what am I saying? I should say that Biostrings and BSgenome do very good jobs of memory management. So in fact, they only load that part of the genome when you request it. It's not entirely all loaded into memory. It takes a lazy approach where it just loads those fragments of the genome that are being used in the analysis at any given time. And when it's not being used, it removes it. That way, all of your RAM on your machine is not taken up, just representing this giant, chunk of DNA. And that's very important. Okay, yeah, it's served, SGD has long served this database as a centralizing force in yeast biology. I can go to it here. This is a nice curated database. So if we go back to our axis of bioinformatics tools, this is definitely not so much a repository and much more a highly curated data set with all sorts of information about this organism and some functionality for BLAST, like we just talked about, that sequence alignment, this guy here, and some things that we're going to talk about later in the course with pathway analysis, etc. Okay. So now I'm going to switch over to RStudio and briefly wrangle with the Yeast genome and annotations and get it into RStudio. And this is going to serve as our third major data set for the third part of the course with computational biology. And we're going to use those three data sets in our analysis. So I think I should show you a little bit about how to get this data in. I find getting the genomes and the annotations to be the hardest part, to be honest. I find it extremely confusing. And then once you get them downloaded to your server, it's generally not very hard because all of these packages have input-output functions to nicely read in the information. There are some points of reflection, but I'm not going to go into them right now. But because they come up, these points come up later as I'm in the RStudio session. Okay, so I'll see you in a second in RStudio. Okay, so here we are in RStudio, Lecture 12, Genome Annotations. All work in R Markdown, show you the code for it. And this should compile to a nice document at the end if I hit the knitter button, but I won't do that right now. I need a little bit of preamble, so the tidyverse and biostrings, and I'm going to need to set my path. So first thing first, because I'm going to download some genomes, I'm going to make a raw directory. That's what I've done here. And I already downloaded the data here, but I would go into that raw directory there. Now, I downloaded from the Saccharomyces genome database, and this is a luxury really because this genome is so well studied and this data website is so well designed and so complete that they make a lot of data available for download. Now, other genomes that are not from model organisms or less studied in general, they are harder to download. That you'd have to go probably to the NCBI, and that can be a little bit tricky. It's not that much more tricky. You might lose an hour trying to really figure out which file is the right file to download. These days, you'll see that the genome and the annotations are made available in different formats, and you'll have to choose which format you want most. Usually GTF or GFF version 3, which is what we're going to see today, is a good choice, but there's personal preference, and almost all of them have functions in our libraries that read them in. Okay, but here, if I go to sequence, I can see I have an option for download, and if I go to download, I see that there's some folders. Now, you really should always read the README files. That's a common good practice, is to have a README file in every directory on Unix so that you know where this data is coming from and where it's going. This README HTML is very short, doesn't say anything really, just that it's Saccharomyces cerevisiae. This sequence README I can show you here hopefully you can see that if I zoom in a bit. It's just basically breaking down what these 4 directories contain. So mainly we're interested in the S288C reference. This is S288C is the most common, I think, lab strain of Saccharomyces. We'll start with that. And if I go into reference, I will see in turn that a series of folders that it's a little bit intimidating. And again, you would have to spend about a good half an hour probably really understanding what each of these files are. I'm not going to go through them all here, but it's definitely where you spend a bit of time. And I can just show you here that open that with my text edit. I'm coming, and just give me 1 second. My text edit, sorry. So it takes a bit of really kind of practice to learn how to navigate all these different file formats. So here is the README folder, and I'll zoom in again. You can see it's explaining one by one, like chromosomes here, the NCBI genome resource at the beginning, the genome release, and then different things, intergenic, ORF, open reading frame, DNA, protein features, and primer sequences, and RNA. So you can already recognize that basically all of the central, the whole central dogma of biochemistry is represented there. And the question is, which one is it that you want to download? If we come in here to the NCBI genome source, here you see that each, let me zoom in, each chromosome is made available under different formats. So this would be a FASTA file, a table, sequence data for chromosome. This is the mitochondrial chromosome, chromosome 16, all the way down to chromosome 1. We don't need that one. There's a simpler way to do this here. If I go to genome releases, I can see that they've made available different releases over time, R9, R8, R7, R6, all the way back. I think the most recent one is down here at the bottom, and this is the one that I downloaded to RStudio Cloud. It's about 16.8 megs, but it's zipped and tarred, so it's going to get bigger. And it's from 2019. Okay, so this S288C current release. And so I would again right click and copy the link address here, and then now this actually contains all the files I need. It's in one package. That's really nice, right? And that's a luxury, right? That's not always going to be that way for every organism. So I would come back here into RStudio, and I would go into my raw directory, and I've actually downloaded this file already. But now, if I hadn't, I would have done wget and pasted the address and hit return, and it'll download that file. Now it has both a GNU zip and a tar compression on it for transport. So I can show you how to do that, but I'll come back up here and I'll make another directory called temporary, go into temp, and I'll download it just to show you. So it's downloading. It's pretty quick. It's not very big, at around 15 megs. It's there, but so now I would do a gun zip of this, and if I do that, I'll see now that my file is called the same thing as before, but .tar. And again, how do you then know what to do with the tar file? I don't. So what I would come up here is, how to untar a file on Linux or on my Mac, on Unix, etc. And I would see that the first hit by Google is pretty clear instructions. It's a pretty common problem people have. So I would come back here, and I know that from literally like a few decades of experience that I use -xf. And when I do that now, I see that this reference is actually this guy here. It's R64. released in 2015. So I guess the yeast genome, the Saccharomyces genome hasn't been evolving, it hasn't, well, not evolving, but it hasn't been changing very much because it's been so well studied. It's the first eukaryote to ever be sequenced, so it's been around for a while. All right, but I don't need this TMP directory, so I'm going to remove it right now. I already have this in raw downloaded, and I can see it there. And this is a directory, so if I go into it, I can see that there's a bunch of files in here. And I can see it better maybe on the graphic user interface. There's a bunch of things here. We're not going to need all of them for today. Annotations clean.gff, I'll explain that in a minute. Gene associations, not feature or coding. These are not features, so they're not genes or anything else. It's all the intergenic, completely, let's say, junk DNA that's in Saccharomyces. The open reading frames for coding regions, other genomic regions like retrotransposons and lines and I don't know lines and signs, but transposons, etc. The coding RNA, and this is the actual this is the big file, 11.8 megabases. That's the genome for that's the 16 chromosomes and one mitochondrial chromosome for the organism. Okay, and yeah, okay, all right. So, whoops, didn't mean to do that. Let me come back in. Sorry about that. I'll get rid of this window here. I would set my raw path as I did before in the previous lectures to cloud project and raw, and then the name that appeared after uncompressing and untarring. Then I'm going to record what website I download the data from. That's good due diligence for the future. And then I start to try and read in the file. I'm going to read in the DNA first. So I know that DNA is going to be in a FASTA format, FSA. That's FASTA sequence annotation. So this is the file I'm looking at to bring in, and so I'm going to use that to upload. So now the question is, how do I get a FASTA format file into R as a biostring? And that's this function called read DNA string set. How did I know that? I had no idea. I went to Google and I typed in something like biostrings read in DNA FASTA, and yeah, right there at the top it says read DNA string set, and that was probably enough to just go and start playing around. So here, not too surprisingly, it takes the file path, which is the raw path up here, this, right, along with the name of the FASTA file here. So how did I know that was a FASTA file? Again, I didn't. I probably would just go in here and say, well, what's FASTA, FSA, DNA, something like that. And well, maybe that didn't really work too well. I might have just asked, maybe said something else like FASTA instead of testing format. And I'm pretty sure, yeah, there you go. I get the first hit is a definition. That's the way it always works. Okay, and in fact, I just specify it should be FASTA. And when I read that in, I'm going to see something that looks like this. Where's my console? Here. Oops, I didn't put this in. Sorry. Initialize this up here. Boom. And now back up and do that. And if I do SC, I can see, in fact, that it has read them in as a DNA string. Yeah, there's 17, which makes sense because there's 16 chromosomes and a mitochondrial chromosome. Now, yeah, there you go. Now, the only thing is that there's some names over here. So I could take a look at that. I could say SC $, and I can see this is how they've labeled the 17 chromosomes. And you'll notice here they've used Roman numerals to represent the different chromosomes at the tail here. And that's really annoying, right? I mean, this is not tidy data, that's for sure. And that's a label. If I just want chromosome, whatever that number 8, I have to type in all of this to get it. That's not really very handy. So, you know, and Roman numerals, why would they use Roman numerals? I mean, you know, it would be easier if they'd used numbers. I'm going to try and clean up the references, the way that they reference the chromosomes a little bit with some code here. So, for example, SC meta, I'm going to call is the metadata. I'm going to take this information, the labels under SC. If I go back here to $, I'm going to take all of this information. I'm going to make its own tibble, and I'm going to chop up that information into bits using the separate function. So here, SC meta, so the metadata about SC, is going to be a tibble, and that tibble is the names from SC. So just to show you here, if I come in here and ask for the names of SC, should work. Yeah, you can see it has these long strings for each chromosome. I'm going to pipe those names. I'm going to make that into a tibble, so I can show you. See how I'm breaking up that long expression to pieces? So now it's a tibble, but it's really boring. It has 17 rows in one column because it has all this, you know, everything is just one character. I can't, you know, if I want to know what chromosome it is or what reference it is, I'd have to do something really fancy to get at that, right? It's not in good computer format. So, and I'll pipe that tibble into a separate, you'll recall from last class. Now separate, the only thing that we have, the only column we have is names, so we're going to separate names into reference, genre, species, strain, etc. So this is reference. This is going to be the genre. This is the species, right? That's genre is the genus, is the parent of the species, and then this is strain, mold type, chromosome, and that's it. And so that should separate that string, and the separator here is a space. So there's a space here, there's a space here between genre and species, etc. And then finally, I really only want, in the end, the reference, which is this guy, and the chromosome, which is this guy. All these guys in here, it's just repeating. We know it's Saccharomyces cerevisiae, etc. Let's just get rid of it and just select down. So I'll show you. And now well, that was a mistake. I don't need to do a double assignment. It's amazing that works. So if I do this, I come in. Now if I look at SC meta, it's a very simple table, but now it has two columns, reference and chromosome, and I have that. That's still really annoying, right? I mean, that's not the way to. It's clearly not a computer scientist or data scientist that made this naming scheme. We should just call these 1, 2, 3, 4 to 16 an M or something. So the way I would do that is I would change that chromosome into a factor first. Okay, so now if I do SC meta, now you can see that it's a factor with those 17 levels. And then finally, I'm going to re-level that factor. And I can do this by accessing the levels. That's a function. I'm asking for the levels of SC $ chromosome. We know what they are, that's these here. But I'm going to reassign them to be the names of SC. Well, really here, this is it. I'm going to rename them as the characters from 1 to 16 and M. So firstly, I'll just show you what this is. It's just, 1 to 16 and M. Now I do a double, I could have done basically twice. I could have said, this and this, but it's just it's in R you can do both at the same time. So basically I've just renamed the columns. When I rename the SC, the names of SC to be this simple numbering scheme and that. So when I execute that, you'll see that now if I look at SC, it just has the chromosomes named 1 to 17. Or 1, yeah, exactly, you know what I mean. And if I look at SC meta, it has a very simple naming scheme for the chromosome. So it's just a bit cleaned up, right? Okay. All right, so now we need the genome annotations. And I'd recommend that you stop the video here for a second and just ask yourself what kind of information annotations might be. I mean, there's genes. And what's a gene? You know, what does it break down into? It's 5', 3' UTRs. It's exons and introns. It's promoters. What else do you want to know about that genome? You have transposons, repetitive regions in the genome. In mammalian, higher order mammalian cells, higher order eukaryotic mammalian cells, like mammalian cells, you'll have enhancers. You might have other regions that are involved in chromatin modification and structure, etc. So that's the kind of annotations that we want to bring into our simple string. I mean, this is interesting. It's very long. This is the mitochondrial genome. It's already 85,000 base pairs long. Chromosome 4, very big. It's 1.5 million base pairs long. But it's kind of useless unless we start to put annotate, layer information on top of it, and that's what we're going to do next. Okay, so now we want to bring in the annotations into our session, and the simplest way to do this for the cerevisiae, in my opinion, is with the GFF file. That stands for General Feature Format. You can read about it more at this Wikipedia link here that you'll see quite nicely in the knitted file. There's GFF, and then there's like GTF, and then there's GFF3, and we're actually dealing with GFF3, so there's been some lineage there. It's a bit confusing. Actually, I just figured it out for this class when I was putting it together. It's really confusing, and I think that I need this package called ape from CRAN. This is some kind of like file format reading it in, and so if I load that library for ape, I think it's been installed in your session by the time you clone lecture 12 at the RStudio Cloud. Should be accessible to you. And I'm going to try to read it in with this function called read GFF. I've never really used it before, but what does read GFF do? There's some help. It's supposed to read in a GFF version 3, and you just basically give it the file name and maybe this GFF3 equal to true. So I tried that. You can see it's pointing to the downloaded file, and when I do that, it errors, and so basically it took about an hour to figure out that it had a GFF. Let me show you the file. It has a GFF file. I don't know who did this, right? But they should be punished. So if I just do a more on this Saccharomyces, this is the GFF file now. When I look at that, you can see that basically it's a pretty simple file. It tells you different features along the chromosome. So here we start at chromosome 1, written in a Roman numeral. SGD chromosome 1, that's a bit better. And this is a position. It goes from 1 to position 230,000. And actually, this entry in the file is basically just saying an event is the entire chromosome. It's sort of like just the whole thing. And it gives you some information in terrible format about that. And then the second event is also on chromosome 1. It's just saying from base 1 to 801, it's a telomere. And then it gives some information about that telomere. It's unreadable here, but that's okay. And then there's an X element. I don't even know what an X element is. I think it's a repeat. It's from 337 to 801. And this file just goes through, and it walks from along the chromosome, each chromosome, telling about each of the events, right? We're up to here. There's an mRNA. I guess the first one is maybe here at position 2,480 to about 300 base pairs or 250 base pairs later. And that's the gene YEL067W. So, basically you can just imagine walking along the chromosome, and then chromosome 2, chromosome 3, etc., and just reading off everything that you see. And that's the GFF file. Pretty straightforward. Yet, when I do that, let me go back and do that, look at it again, I figured out that halfway through the file, they switched and they put the genomes, like the actual FASTA file, for all the chromosomes. So I cannot understand for the life of me why they would put a GFF file at the beginning of all the events, all the annotations, and then they put the chromosomes at the end. So I had to script them, and I made a new file called annotations clean, which you'll have access to. And this is just the GFF part and not the FASTA raw genomic data. We already have that in, we don't need it anymore anyways. And so now when I read in the annotation file, you'll see it's now the cleaned one. When I read that in, whoops, I'm not in my R session here. There. Now TMP, well, you know, it's big, right? It's a data frame. Let me see if I can just back up and give you, it's not a tibble yet. So you can see now, that looks like a GFF file. Again, the first event is the entire chromosome, and then the telomere, that X telomere, the first gene is down here, etc. So that works now, right? That took nothing to get into R. Now the problem is that the annotations are terrible. So I'm going to show you here. I'm going to do three things. I'm going to make it, call it anno, and I'm just going to make that TMP into a tibble because we like tibbles and they're a bit easier to work with. And then I'm going to remove some of the big data structures that are there. They're just taking up space in memory, and we're not going to use them anymore. We could just ignore that error there. Okay, so now if we look at anno, it'd be a bit nicer to look at. You can see it looks pretty good actually, and I think it's, these columns are tidy, but then you get to this nonsense. Like this is just, I don't know for the life of me why they would choose to just use one character, right, to represent all of this information. So, you know, you'd have to go in here and pull out chromosome 1, the reference number for the NCBI, and then, you know, and then there's all sorts of different events that are encoded in there. That is not tidy data, right? That is really a disaster, and there's no easy way to parse that information out. I didn't parse it out because I think it would take really quite a while to do. But, and I'm not sure we need all that information. Although, you know, we have that it's a gene here, but we don't know the gene name. So this first gene on chromosome 1 is really just past, you know, it's right at the end of the telomere. It's called YAL069W. And we'd have to go and dig that out of this line here in the attributes. I wrote a function that takes one row of our tibble, one annotation, and it searches this attributes field for a specific annotation and returns that. Let me give you an example. This is anno, and if we look at anno 1, okay, this is the attributes field. And suppose I wanted to know the dbxref for this first entry, which is NCBI. That's the answer is this, NCBI, NC number. Okay, that's the answer. So I want to write a function that I could say, look in row 1 and look in attributes for the dbxref and return the answer if it exists. If it doesn't exist, it's going to return null. So let me give you an example. I'm going to copy and paste this function that I wrote in, and I'm going to look and say, now the whole thing is going to be in anno 1, for example, and that's what that is equal to, okay? Those three different, let's call them fields that are within that, not that one, this name, dbxref, and ID. So if I run this guy, find attribute of the first row of ID, I get back CHR1, right? Chromosome 1. And if I ask for the dbxref, I get back NCBI, NC. Name, I get back chromosome 1, 2. And you can verify that's correct. And what if I ask for a field that doesn't exist, like nonsense? I get null. So the function works as it's supposed to. Now, how does it work, right? Okay, so let's suppose our attribute that we want to find is set equal to dbx ref, okay? And annotation is going to be the first row. So that's going to be anno 1 comma, right? That's 1 comma because it's the row 1, and if I don't put anything beyond there, it takes all the columns. So it's the entire first row, okay? And now I pass that, that's what's passed to my function. So target now is going to be assigned equal to the character version of annotation attributes. So what does that look like? I'll just copy and paste this. See, target is just that full string, right? Now our goal is to get this guy, right? That's what we want to return, just that. So the first thing is to find dbxref equal to. And the way I do that is I use the string locate from tidyverse in target, that string above, and I use the string concatenate, C, of attribute and equal sign. So I'll show you what that looks like. That looks just like name, whoops, what happened here? My attribute is dbxref. Did I make him, oh, I made a mistake here, I'm sorry. Attribute, I missed it here, I didn't see that I had an error. dbxref, okay? Now when I come in here, this should look like dbxref equal to, and it does. So that's, I'm going to use that to find this. And now I use string locate to find in target this string here. And when I do that, what I get back is a pair of coordinates. Index looks like this. It says it starts at 9 and ends at 15. So if I count 1, 2, 3, 9, and I look at the end, which is 15, that's going to be the equal sign. So that says the start and end of that motif, that sequence, right? Okay, so I found that. Now I want to dig out this part. So the first thing I'm going to do is just take everything to the end. So I come in here, and first I'm going to ask, did I find something? Is it NA? And that's going to be no, right? Because it did respond with actual indices, so it found it. So it's not true. So I come to the else clause. Now, TMP is going to be equal to everything from here all the way down to the end. I'm getting there, right? I've got this, but how did I do that? Well, I used string substring on target again, but now I used index 1, 2, plus 1. So index, remember, index at 1, this guy, index 1, 1 is the start, but I want the end, right? I want 15, because that's where this equal sign ends. So I want that plus 1, which is going to be exactly this capital N. And how long do I go to? In this case here, I go all the way down to the end of the string. I just take the string length of target, and that's going to be all the way down here. So when I run that, I see it's got all of this. So I've made a bit of progress because I've gotten rid of this part. That's gone. Now I need to get rid of this part, right? Okay, so I come in here, and now index 2 I run, and I use string locate again on TMP, this guy here, right? I use on TMP, I'm sorry, I'm right here. Index 2 is run on TMP, and I look for $, and this is a regular expression. And this means I look for the first time that I find a semicolon, and that bar means or the end of line. $, means end of line. So when I run that, I see that index 2 is right at the end of that sequence, right? So let me show you TMP again. So if I count through here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 is right there, okay? That's exactly what I want, right? So now, is it null? No, it won't be null because it actually found numbers, so it's false. So I don't return anything. Instead, I come down here, and final now is actually equal to the answer, right? NCBI, etc. And so here I can see it on TMP, that's this string. Let me show it again. Oh, TMP is right here, okay. I go from 1 to the index 1, 1 minus 1. That takes a bit of explanation. So index 2, recall, is 15 at 15, right? It's pointing right here. So I don't want that, I don't want the end, right? I want to get rid of it. I want to stop one character before. So no, I subtract -1. And now final is the answer, and I return final from my function. And that's exactly what you get, right? You get that. Now, if I asked for name, well, let's say nonsense. Okay, so let's go through this for nonsense, and we'll end there. Annotation is still the same as before. We don't need to change that. But attribute now is going to be equal to nonsense. It just doesn't exist, right? So let's go through it. We come in here. Target starts off at being all of attributes, right? Nothing's changed. And now I run index, and index is equal to NA. That's because string locate in target didn't find attribute thing. So nowhere in there was there nonsense equal to, and it gave back NA here. So when I check here, is it NA? It says true, and I return null. Okay, there's one other, and then, the other case is that it's an interesting exercise for you to go through with ID and name and see if you understand how that works. And there's a little bit of code down here that will print out everything across ID in a for loop for every row of your tibble. And I think there's a lot of rows, right? There's 23,000 rows and annotations across the yeast genome. But now we have everything there in R, and we're ready to do lots of nice analysis on the genome. I'll just finish by saying, we can easily investigate a gene now. Let's take this guy here. Now, if we want to see that gene, we can just come in and say SC. We're on chromosome 1, right? And we want to look at 335 to 649. And that's the actual sequence for that gene. And now we could do all sorts of interesting analysis. We could ask for the promoter region, let's say 100, let's say maybe 200 base pairs upstream of that gene. And we could then say, instead of from 335, we could say maybe, I don't know, 135 to 335, and that should be the promoter for that gene, right? And etc. So now later on, next class, we're going to stay with Saccharomyces, but we're switching to part 3 of the course, and we're going to start to look at analyzing the promoters of these genes for transcription factor binding sites. All right, guys, I hope that that was clear and you got something from it.